Fine. See, in today's class, we will discuss about DNA. Why is it a genetic material? Some evidences we require. In previous class, I told you about Hershey and Chase, how they utilize radioactive nucleotides and radioactive proteins to prove that DNA is a genetic material. What exactly they did was Hershey and Chase, they took up the bacteriophage, which infects a virus which infects bacteria. We call it as bacteriophage. In one of the bacteriophage, what they did was the protein capsule that is there, it had the radioactive sulfur, 35 sulfur. This is radioactive and it can emit radiations. Okay, so one of the bacteriophage was exposed to they had this radioactive sulfur, and another bacteriophage, they had this radioactive phosphorus. Why did they utilize sulfur and phosphorus? Why not any other compounds like carbon, hydrogen? They are found commonly in proteins as well as in case of the nucleotides. Okay, so they wanted to have certain materials which are unique to the proteins. Sulfur is found in most of the amino acids. Okay, so sulfur is unique for the protein part of it, and phosphate group is an essential part of DNA or RNA. You should understand that it is a part of nucleotide. Okay, sugar plus nitrogenous base plus phosphate group. That is what we call it as nucleotides, isn't it? So that's the reason they chose phosphorus, which is radioactive. 32 phosphorus they have chosen here. So the bacteriophages which has 32 phosphorus, radioactive labeled DNA, and here it is only the protein which has radioactive 35 sulfur labeled protein capsule. Why should they have it as radioactive label? What is the purpose of it? Okay, they can easily trace it because radioactive material they emit radiations, and you can trace it out where it has gone. In order to trace this, they use this radioactive nucleotides. That is the purpose of this experiment. So Hershey and Chase, they did that. Okay. So now this bacteriophage, they infected bacteria. That is a bacteriophage with radioactive protein capsule, sulfur 35. Okay, this protein capsule they infected it. Infection stages, if you look into a bacteriophage, they have the neck, the tail region, basal plate, tail fibers are there. So they come and fix to the bacteria, and they are going to introduce this during infection, the genetic material from the bacteriophage or virus, they enter into bacteria. They are injected them into this. The mechanism and process will not discuss about it. This DNA that has been injected, they take control, they destroy the bacterial DNA. They take control of the protein synthetic machinery of the bacteria. It is almost like they have hijacked the bacteria. They have killed the pilot. What is the pilot here? The nucleic material of DNA. The nuclear material of DNA has been destroyed, or the bacteria has been destroyed. Instead, now they have the material, genetic material of virus. Now the virus, it takes control of the protein synthetic machinery, and they are going to synthesize a large number of such bacteriophages within them. Then the bacteria bursts open and they releases this bacteriophage. This is what happens in the cyclic life cycle of a bacteria. Okay, so now they have injected this, they take control of the protein synthetic machinery. Now the bacteriophage without genetic material, what do we call it as? We call it as ghost. They are subjected to centrifugation. So supernatant character, but generally they are ghosts. Ghosts are without bacteriophages, without genetic material. Whereas the genetic material one, it has been injected into the bacteria. Okay, remember about that. So now they are liberated. The ghosts are liberated, and you know that the ghost or the protein capsule is outside. So they are having this radioactive nucleotide sulfur. Okay. Now this genetic material does not have radioactive nucleotide. So you can notice that this ghost will be emitting radiations, not the radioactive nucleotide. If this radioactive protein capsule capsule was the component which infected the bacteria, then it should be inside. But it is not found within the bacteria. There is no radioactive nucleotide sulfur within the bacteria, which indicates that protein is not the genetic material. Protein doesn't cause the infection. Okay. The second step is blending. They blend, and they are going to utilize the protein synthetic machinery. Now, when you centrifuge, which is the third step, centrifugation, when you do that, you find that there is no radioactive 35 sulfur detected in the cells, bacterial cells. When I am talking about cells, I am talking about bacteria. But the radioactive 35 sulfur is detected in supernatants. Supernatants contain this protein code. That is why I have given it as a yellow color here. They are radioactive nucleotide. Sulfur is there. They are found in the ghosts of the bacteria. So now from this experiment it proves that protein is not going to be the genetic material. Now in the next experiment what they have done, the bacteriophage is having the DNA with radioactive phosphate. That is 32-phosphate is found in the nucleotide. So radioactive 32-phosphorus labeled DNA is found in this bacteriophage. Infection stage is similar. They inject the genetic material. The genetic material is injected. They destroy the bacterial DNA, and the viral DNA hijacks the bacterial cell, and now they start undergoing multiplication. This viral DNA, they produce large number of bacteriophages. Once they are very large, this bacteria bursts open, releasing this bacteriophages, which infect new bacteria. That is what is the life cycle of bacteriophage. We are not going into that. Our concern is to see what happens to this radioactive labeled phosphorus present in the DNA that has entered into the bacteria. This radioactive labeled DNA has taken control. This is viral DNA. It has taken control of the bacterium protein synthetic machinery. They produce large number of copies. Now, when you subject this sort of after infection blending, when you subject them to centrifugation, the host does not have, the supernatant does not have any radioactive component. Okay, the protein part does not have any radioactive component, but the bacterial cells, radioactive 32-phosphorus detected in the bacterial cells. What does it indicate? DNA, since it is the genetic material, it has entered into the bacteria. So now you can understand that DNA is a genetic material. If it was not a genetic material, it would not have entered inside the bacteria and destroyed the bacterial DNA and taken control of protein synthetic machinery so that they can synthesize their own copies. It is just like an aeroplane being hijacked by any of the terrorists. The viral DNA is going to hijack the bacteria, and they are going to utilize the protein synthetic machinery to produce their own copies. Okay? That is what is going to happen. Now, when they centrifuge, they found that the radioactive 32-phosphorus was detected in the bacteria, and the supernatant did not have any radioactivity, which indicates that the DNA is the genetic material, because phosphate group is found only in the radioactive phosphate group is found only in the DNA. This is what you have to understand about Hershey and Chase experiment, which provided a very concrete evidence to prove that DNA is the genetic material. DNA is the genetic material. That is what this entire experiment is about. Now, properties of the genetic material. You can also call this topic as DNA versus RNA. In majority of the organisms, whether it is in plants or animals, DNA is the genetic material. Very few of them, like a few viruses like HIV, human immunodeficiency virus, tobacco mosaic virus, here they have given a QB bacteriophage. They are all having RNA as a genetic material. Then you have a question, why shouldn't RNA be the genetic material in all the organisms? Why does nature prefer more of DNA over RNA? What is the cause for that? Why is DNA more of the genetic material than the RNA in majority of the organisms? And for any molecule to be a genetic material, what are its attributes or properties of genetic material? What are the characteristics of genetic material? So that is what we will be discussing in this topic. Okay, I've already told you few viruses we are having RNA as a genetic material, like TMV, polio virus also. Poliomyelitis virus also has RNA as genetic material. HIV, I told you, human immunodeficiency virus, sarcoma Rouse virus, QB bacteriophage, all of them, they are having RNA as a genetic material. And if they undergo transcription, what do we call it as? Reverse transcription. RNA synthesizing DNA, we call it as reverse transcription. The synthesis of DNA by such reverse transcription, we call it as cDNA, or we also call it as complementary DNA. But now you should understand a few of the organisms like viruses, they are having RNA as a genetic material. Okay, but DNA is predominantly genetic material in majority of the organisms, isn't it? So that you have to understand about. Now RNA has dynamic functions. They are acting as a messenger RNA as well as adapter RNA. If you look into the central dogma of molecular biology, DNA produces its own copy by replication. So that is one first process in central dogma of molecular biology. Second, DNA synthesizes messenger RNA, which has the codes. We'll discuss about that genetic code later on. mRNA in turn, they undergo translation or protein synthesis to produce functional proteins, which can be either hormones or enzymes. Functional proteins are the protein. Okay. So that is the central dogma of molecular biology. Replication, transcription, translation. These are the three steps that are involved in. So now RNA dynamic functions are there. It is going to be acting as a messenger RNA. It plays a major role in synthesis of protein. In the expression of gene, RNA plays more of a major role than DNA. DNA just has a message. It is a storage of genetic material. But this transmission of this gene or expression of gene is brought about by RNA, especially mRNA and tRNA. Messenger RNA, adapter RNA is nothing but tRNA. When we come to protein synthesis, I'll discuss about that. So the molecular genetic material, if you have to call any molecule as a genetic material, 4 criteria ಬೇಕಾಗುತ್ತೆ. 4 criteria are required. First, it should be able to undergo replication. It should be stable chemically and structurally. Chemical and structurally, it should be stable. ಮೂರನೇದು mutation ಆಗಬೇಕು. Any sudden change in the structure or genes, we call it as mutation. Okay? Any sudden change in the structure or sequence of genes in a chromosome, we call it as mutation. Okay? So mutation ಆಗಬೇಕು. ಮೂರನೇದು, it should express characters according to Mendelian principles. Law of dominance, law of segregation, law of independent assortment. They should follow these principles. ಈ 4 criteria ಇದ್ರೆ ಯಾವುದೇ ಒಂದು ಮಾಲಿಕ್ಯೂಲ್ ತೋರಿಸಿದ್ರೆ ಅದನ್ನ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ ಅಂತ ಕರೀಬಹುದು. ಈಗ ನಮ್ಮಲ್ಲಿ ಜೆನೆಟಿಕ್ ಮೆಟೀರಿಯಲ್ DNA ಅಥವಾ ಕೆಲವು ಆರ್ಗಾನಿಸಮ್ಸ್ ಅಲ್ಲಿ RNA ಅಂತ ಹೇಗೆ ಡಿಸೈಡ್ ಮಾಡೋದು? By this 4 criteria. First, it should be able to undergo replication of its own copy. Second aspect, it should be chemically and structurally stable. Third aspect, it should have mutations. Mutations or variations are raw materials for evolution. Mutations and variations are raw materials for evolution. And they should undergo Mendelian characters or principles of Mendelism should be shown by these molecules. If they do that, then you can call them as genetic material. Okay. Now, what you can notice now, DNA versus RNA. Let us look into that. DNA can also replicate. RNA also can replicate. Both DNA and RNA can replicate because of rule of base pairing and complementarity. What is complementarity in RNA? Adenine always base pairs with uracil. The bond would be double bond or vice versa. Cytosine base pairs with guanine and the bond will be triple bond. Even RNA has that. So since it follows both DNA and RNA can undergo replication because it follows this base pairing, rule of base pairing and complementarity. Instead of trimethylene you have uracil. But thymine is more stable than uracil. It will find further thing. Thymine in DNA is much more stable than uracil. So that's the reason DNA is more stable than RNA. Next point will come for that. So proteins fail to fulfill this criteria. Proteins do not have base pairing, neither do they have the complementarity. That is the reason proteins are not genetic material as per Hershey and Chase. Next, genetic material should not change at different stages of life cycle. Like when you are infant, the genetic material is active, but when you grow up, it should not become inactive. Throughout lifetime, genetic material remains stable. Life cycle only, age only, and change in physiology, DNA does not get affected. DNA remains the same. That is what is that point. Genetic material is stable in different stages of life cycle, age, or in physiology of organism, which is visible in Griffith's transformation principle. Griffith's experiment that is heat killed a strain of the Streptococcus pneumoniae. Okay, heat killed a strain of that bacteria. Even though the bacteria dies, the genetic material is alive. Heat gain ಮಾಡುತ್ತೆ ಆ DNA ನ ಡಬಲ್ ಬಾಂಡ್ ಇರೋದು ಆ ಬಾಂಡಿಂಗ್ ಬ್ರೇಕ್ ಆಗಿರುತ್ತೆ. They become single strands. Okay, heat can just break the double bonding or the helical structure of the DNA, but it cannot destroy DNA. So, Griffith transforming principle experiment, if you observe, the bacteria is killed, but the DNA remains active. The heat that was provided to kill the bacteria, it is just going to break the DNA into single strands. This single strand, on favorable conditions, they can base pair with complementary strands. Okay, that is what you notice when our strain of bacteria was introduced. They entered into our strain and they found complementary base pairings. So that is how the R strain of this bacteria was transformed from A virulent to virulent A strain by this DNA. I experiment on previous class did. That itself is a very good example to show that stability in DNA is more. Even heating them, the bacteria gets killed, but not the DNA. That is what you have to remember about. Only DNA strands get separated by heating, which come together when appropriate conditions are provided. Andre base pairing ಆಗುತ್ತೆ. R strain ಅಲ್ಲಿ appropriate condition ಇತ್ತು. ಈ S strain DNA ಏನಾಯಿತು? It became double bonded, double helical structure ಆಯಿತು. They converted or transformed this R strain into S strain. That is why they became A virulent to virulent, and they caused the disease when there was a mixture of heat killed S strain plus R strain. Okay, I will then before the fourth experiment that was conducted. Then 2OH group is present in RNA. In every nucleotide of the RNA, it has that 2OH group. What is it going to make? It makes the RNA reactive. They are very volatile and they are very reactive and makes DNA labile and degradable. They can be easily degraded. But DNA is agalla. Okay? RNA is catalytic in function. Thus, DNA is chemically less reactive and more stable when compared to RNA. That is why nature prefers DNA which is more chemically stable than RNA. RNA, since they have that 2OH group, they are very reactive. Okay? And they can be easily degraded. That's the reason nature prefers DNA over RNA. Okay, now another point for DNA to be much more stable is thymine, which is present in DNA. RNA has uracil. Instead of thymine, they have uracil. But thymine is much more stable than uracil and they provide stability to DNA. Whereas uracil doesn't do that job to RNA. That's the reason DNA is much more stable. Okay, and it is much more preferred genetic material. Both DNA and RNA can mutate. Mutations can take place, but RNA can undergo more mutations because they are very much reactive. RNA is more reactive because of the 2OH group. Mutations ಸಾಧ್ಯತೆಗಳು RNA ನಲ್ಲಿ ಜಾಸ್ತಿ ಇರುತ್ತೆ. DNA ಗಿಂತ RNA ನಲ್ಲಿ ಜಾಸ್ತಿ ಇರುತ್ತೆ. So that is one thing. Both can undergo mutation. Okay, this is a fight between DNA and RNA. But ultimately who wins? DNA because of various attributes. Okay, both DNA and RNA can be a genetic material, but DNA is more stable. So nature prefers in majority of the organisms DNA as a genetic material. DNA, what is it going to do? Without RNA, it cannot express its character, genetic character. DNA is storage of the genetic information. What is DNA? It is just like your pen drive or RAM, memory, CD-ROM or hard disk idange. It stores genetic information. RNA expresses the genetic information by transcription and translation. That is, transmission of genetic information is brought about by RNA. This is the conclusion of it. In the next class, we will discuss about RNA world.